All right, welcome everybody. So every year I get the distinct honor and privilege of presenting the MCHRI Visionary Leadership Award. It is so fun for me because I always know about the work these amazing visionary leaders have done, but in the process of preparing the remarks, I really get to do a deep dive, as David will probably remember from last year. So this year, it is my great pleasure to present the MCHRI Visionary Leader Award to Dr. Virginia Winn, Professor of Obstetrics and Gynecology in Reproductive, Perinatal, and Stem Cell Biology Research. We've already heard a little bit this morning about the importance of preeclampsia, and Dr. Winn's laboratory seeks to unlock the complex biology of the human placenta and determine how its dysregulation leads to obstetrical complications. Her work spans the spectrum from basic cellular and molecular biology of the placenta to machine learning prediction models of preeclampsia to translational clinical studies. Her lab has established cutting-edge stem cell model systems both for the trophoblast and endothelial cells, and I could tell you a little bit about the basic science in each, but I'll just do a high-level overview, and they're advancing studies of the placental and maternal contributions to the pathogenesis of preeclampsia. These model systems enabled the identification of preeclampsia-associated molecules and exploration of their role in preeclampsia pathogenesis prediction and therapeutic strategies. And then in related work, she and her team demonstrated that the maternal endothelial cell function and endothelial progenitor cells are impacted by the hormonal environment at the time of conception based on using a natural cycle or a medicated cycle for frozen embryo transfer, and this important discovery likely explains in part the increased risk of preeclampsia with IVF. Looking back through some of our records, in 2015, she was given one of MCHRI's really competitive, wonderful faculty scholar endowments. So in 2015, she was named the MCHRI Arlene and Pete Harmon Faculty Scholar for her project entitled Endothelial Dysfunction in Preeclampsia, Implications for Immediate and Long-Term Health Outcomes for Mothers and Children. So fun for us in terms of just how MCHRI supports our faculty, Virginia, to look back, see that you got that faculty scholar in 2015, and then to see everything that's happened since then. So MCHRI will claim some credit for that, because that early work and those projects established the scientific basis for really two very landmark, prospective, innovative clinical and translational studies focused on different aspects of preeclampsia. So I want to tell you a little bit about the EPOC study and then the NAPPRO study, because I think they're both beautiful demonstrations how our basic science informed clinical research and then really clever study design. So first is the EPOC study, which she co-leads with Mark Ladke, is a large NIH-funded team science project to define the molecular basis of the effect of preeclampsia on later cardiovascular health. So what they did in a really clever design was to have 3 separate cohorts, and then they could sort of stitch together the whole life course across these 3 cohorts. And it's been really fun because just in the last few months, 3 great papers have come out of this study. So what they did is they took a life course and multi-omic approach to tackle this question about the effect of preeclampsia on later cardiovascular health. They used 3 well-defined, well-characterized ongoing cohorts of women. So the Stanford March of Dimes study of pregnancy, the Danish National Biobank, and the Women's Health Initiative. And then with extensive cardiovascular, proteomic, metabolomic, lipidomic, immune profiling, endothelial progenitor cell and placental transcriptomic studies from pregnancy, and then in these later life cohorts, gave great insight into the omic signature of preeclampsia, its resolution and persistent differences years after delivery that may link to cardiovascular disease. So the publications are rolling off the press. I had to email her 2 days ago just to make sure I had all of them. And one of them, they assayed over 7,000 plasma proteins. They applied machine learning to identify the proteomic signature at each time point and then performed enrichment analyses to identify relevant biologic pathways. The pathway analyses identified differences in the complement and coagulation cascades that persisted after the antepartum, that actually persisted across the antepartum, postpartum, and midlife samples, and differences in protein expression persisted for years, decades, after the hypertensive disorder of pregnancy. And it was the consistent differences in the complement and coagulation pathways may contribute to the increased risk of later life and cardiovascular disease. So incredibly impactful work and so beautifully aligned with what we're talking about today with maternal and child health and then the life course. The second study has been also great fun to watch. This is a study she leads with Ruth, who was our first speaker today. This is an NIH-funded randomized clinical trial that assesses how the method of frozen embryo transfer, either natural versus program cycle or NAPRO, impacts the development of preeclampsia and live births. The study, this goal was to enroll 780 women, and they did it. I think that's all the more amazing coming out of a pandemic. And this really has the potential to decrease the incidence of preeclampsia for IVF pregnancies by changing clinical practice overnight. The data collection is nearly complete. The last delivery is expected this fall. So we're really excited to see what that shows us. But as a physician scientist, remembering that these amazing studies are founded in her fundamental discoveries, she really wants to understand the why that underlies these clinical observations. And she's already initiated a number of ancillary studies to NAPRO funded by MCHRI, the Cardiovascular Institute, and the Women's Health and Sex Differences in Medicine to assess how the mode of frozen embryo transfer impacts the maternal immune response. So we look forward to all that work as seeing how that unfolds. Now, Virginia has been sort of our leading placental biologist on this campus for a long time, and she's served as the placental pregnancy or preeclampsia expert for many other research projects here at Stanford. These include investigations with the March of Dimes Prematurity Center, focusing on the molecular underpinnings and prediction of preterm birth and preeclampsia, collaborations with colleagues in RNAi genetics on the placental and maternal contributions to recurrent pregnancy loss with the TRIOS and HOPE study, and she serves as the site obstetrician on the Sunbeam study, which is the largest pregnancy cohort study to determine perinatal and environmental contributions to atopic disease and food allergy development in children. Virginia is a very gifted and generous mentor. During her time at Stanford, she's mentored many high school students, undergraduates, and medical students. I've learned that the team of undergraduates who collect placentas and cord blood for her many translational clinical studies are part of what's known as the placental posse. And she has multiple former trainees that are now accomplished faculty, and we're also so grateful that she has served as a mentor for our DRIVE students program that I mentioned in my opening remarks. In 2020, she became the program director for Stanford Women's Reproductive Health Research, or WRHR, K-12, and the principal mission of the program is to provide state-of-the-art mentored multidisciplinary research career development for outstanding junior clinician scientists who will impact and improve women's reproductive health. Turning to MCHRI, she's been an extraordinary contributor to this institute. She served on the education committee for 5 years. We were so grateful to have her on the executive committee for 8 years. And I want to tell you the story of how Virginia was the motivation to change our name. So when Mary Chen developed our logo, up there on the top left, I heard Virginia mutter under her breath, well, at least the mother's in the logo. And I said, oh, let's change the name. And right there, that day at the exec committee, we voted to change the name from CHRI to MCHRI, and the rest is history. So thank you, Virginia. It was so obvious once you said it, but somebody needed to say it. So thank you for that. She also has had a really important role. Our clinical research support office, I said a little bit in the opening remarks about how we enable research within the Children's Hospital. And because we are a mother and children's hospital, we really needed that voice on the executive advisory committee. And then another real labor of love, I hope you all know how much we treasure the work we do to fund our postdocs. And Virginia chaired one of the postdoc study section panels for 5 years. In summary, she's a nationally and internationally recognized for her expertise in placental biology and preeclampsia. She was elected to the Leadership Council for the Society of Reproductive Investigation. We are so incredibly proud to present this award today, and so she can add MCHRI Visionary Leader to her long list of accomplishments. So please join me in congratulating and thanking Dr. Winn. Okay. So, Virginia, come on up. We started a new tradition last year. We did something a little bit different. I want everyone in this room who's Virginia who has touched, because you were inspired by her, because she mentored you in any way that Virginia has affected your career. I want you to come join us for this photo. So all of you, come on up. Yes, good. All right.
What are you doing?
In my lab, when we take a picture, what do we say? 1, 2, 3. Placenta. There you go. That's what we used to.
Great. Well, thank you, Mary, and thanks to everyone for this incredible honor. When I was notified to be the recipient, I was really humbled to be in the company of those that have received this honor in the past. And quite shocked and very, very appreciative. I actually also want to thank MCHRI because I have seen firsthand the impact it has on my science, my career, my ability to mentor the next generation of reproductive scientists and clinicians. And I was recently in Santa Fe, and just north of Santa Fe on the mountains there is one of the largest aspen groves. And this time of year, they're gold and the leaves shimmer with this brilliance, and it's just an amazing sight. And when I was reflecting on what MCHRI has meant and been for me, is an incredible privilege and honor to see the impact it makes on this field. And in thinking about the aspen, if you don't know, have a root system that connects that entire grove. And what MCHRI means to me is it really helps get that root system complex, networked, and extends that grove into areas that you wouldn't necessarily anticipate, and provides the opportunity for those young saplings of aspen to shoot up, be strong and tall, and then to glimmer with incredible impact in the fall with the gold leaf colors. So thank you to MCHRI. It's been a real privilege to be part and see that growth and impact that will continue well beyond where we're at. And I look forward to years to come of being part of this amazing community. Thank you again so much.